My name is Tuulia Huhtola, and I'm the head of biomarkers and molecular imaging here in Charkivi, Finland site. I have a background mainly in nuclear imaging, but now I'm been also leading the biomarker labs. So our biomarker lab is known especially for the histology, and that is one of the key components what we are adding for pretty much all of the studies what we are doing. We do both traditional histology but also immunohistochemistry. Then in addition to that, we do a lot of protein analysis, so we can do both protein concentration, activity, kinetic measurements and that type of things. Then we also do a lot of gene expression analysis, for example using branch DNA technology. So it's very cost efficient, fast, reliable and sensitive way to look at many different targets from same sample. We also do flow cytometry, what we apply, for example, blood analysis of different cytokines and inflammatory markers of samples. We can process for the histological work. We do all the processing by ourselves in our facilities, and meaning that we can handle both paraffin samples and as well cryosections and cryosamples. We do both of the microtome and cryotome sectioning in-house here in our facilities. For the histological detection, so we do both colorimetric stainings as HE, but we do also a lot of immunohistochemistry, where we can do detection of the primary antibodies both using colorimetric dyes or then also fluorescent labels. So we have microscope in-house, including all the channels for blue, red and green. So we can nicely quantify the events of different channels. So it also enables us to have three different markers at the same time from the same section. Autoradiography is very old methodology applied in many disease areas from traditional biodistribution studies. So it's very nice way to combine also with histology, traditional histology. So we can have several set of sections from the same area of interest, and then look detailed about the ligand binding, what is the receptor level. The other thing where autoradiography can be widely used is receptor occupancy assays, where you can compare the novel compound binding and then competitive binding to the known molecule to the same receptor target. In vivo results give very good information, for example, behavior or changes in the phenotype or that type of things. When we want to look more detailed about the actual events and biomolecular level, that what type of changes there are in different tissues over the disease progression and whether the treatment has had any type of effect, it's actually the biomarker analysis which then kind of confirms that there was, for example, inflammation or the neuronal loss. Or, for example, inflammation is very interesting topic at the moment for disease progression studies and also for the drug development progress. So the inflammation is related to so many disease areas, and that's very typical marker. For example, multiple sclerosis disease models. We can look at, for example, the using flow cytometry and different cytokine panels and different cells related to the inflammation process from blood. In addition to that, we can look at cytokines. We can use Luminex assay to look up to 30 and even 50 targets from the same sample, how cytokine concentrations are altered or upregulated in the disease animals. Where we can help with our components for the client studies is really that we can measure the actual changes in biomolecular level and what type of pathology there has happened and whether there has been effect on the compound treatment.